part 2 of chapter 4. So we're going to be focusing on eukaryotes in this particular section. So as we get into it, let's talk about the cell membrane. Now this is true, of course, for prokaryotes as well, but we're going to be focusing on eukaryotes. But the cell membrane, this is the living boundary, the boundary between the living and non-living thing. It's you have a living cell, that's the edge, that's the boundary of it. That's what separates the cell from the world around it. Everything in the cell membrane is the cell. Everything outside is not. There are things the cell produces that go out. It's what separates the cell from its surroundings. Now, very important about this is it's selective. Now, when we say it's selective, I like to think of it this way. It's kind of like people in school when they're dating, and you got some people, they'll date anybody. Then you got some people, they will not date anyone. But you got people in the middle, and they're picky, but they will date. If you are their type, they'll date you. You meet my criteria, I will date you. Okay, we're not saying their criteria is always the best, but maybe their criteria is a little bit too picky. Maybe they're a little too superficial, too shallow. But maybe they're like, hey, as long as you got a job, or as long as you treat me right, or as long as you're a good person, things like that. So we look for characteristics, and we say, if you are that, then I will let you into my life, and they'll date you. Okay, well, that's kind of what cell membranes do. Is they are selective, they are picky. They let some things in, some things not. Okay, some things out, some things not. What can go through a cell membrane very easy is small nonpolar molecules. If you're small and you're nonpolar, you go through very easy. Why is that? The cell membrane is mostly phospholipids. The phospholipids let certain things in and certain things not. Okay? So because the cell membrane is made of lipids, small nonpolar things go through easy because they are themselves not charged, so they're not lipophobic. They're lipophilic. Okay? Now, so the phospholipid bilayer is what the cell membrane is made up of, is a bilayer of these different lipids. Okay? They're hydrophilic phosphate heads. That means that part likes water, the heads. The so-called the little balls, the yellow balls here, those are the heads. And the blue tails, they are hydrophobic fatty acid tails. So we refer to this as amphipathic in biology. Now, they're peripheral proteins, and they are proteins on the periphery. They are on the inner and outer surface of the cell membrane. Then there are proteins that poke through the cell membrane called integral proteins, and they are integrated. They are poking through it. They pass through or span the cell membrane at least once. Most of your peripheral proteins are enzymes. Most of your integral proteins are transporters. Glycoproteins, they are attached proteins to a sugar. They will have some carbohydrate sugar. Glycolipids are lipids with a sugar. And what these do is they lubricate the cell. That's that glycocalyx we talked about. Okay? Now, there are molecules, lipids and proteins are the major part of it. What we kind of talk about is that there is this ocean of phospholipids and these proteins that are in it. So by far, phospholipids is the most abundant lipid. Okay? Then there's a mosaic. What do I mean by a mosaic? I mean, so have you ever seen those pictures where they take broken pieces of tiles and they smash a tile up? Then they take these pieces of different colored tile and arrange them into a picture. It's called a mosaic. Well, this mosaic that we have is a fluid mosaic. We have a fluid cell membrane with proteins bobbing in it like a mosaic. We call that a fluid mosaic model. Okay? Now, randomly distributed, I don't like the way that, I always feel like there's some things always left out of our book, and I always want to kind of fill in the gaps with my own self. But anyway, now what happens is, these integral and peripheral proteins, let's talk about them a little bit more. I always talk about integral proteins, they penetrate the hydrophobic interior. So they may go all the way or some of the way, part of the way through. They are called transmembrane proteins. That's because they can allow passage. They oftentimes are channels. Then there's the peripheral proteins they don't pass. They're loosely bound to inner and outer surface. They're usually enzymes. Okay. Now we talked about how the cell membrane, that it's an isolator. It's a barrier, but it's not a perfect barrier, but it does physically isolate. So what's outside and what's inside are different. There's different worlds going on there. Different chemistry inside, different chemistry outside. So one of the things we can know as well as is it regulates the environment. It's like a border, okay? If you want to go cross a border, you go in, cross the border, and they may say, do you have anything to declare? What's your purpose? State your business here, da-da-da-da, yeah. Oh, I'm here for business or pleasure, whatever. And okay, and they let you in. Okay, we need, okay, good. Come on in here, spend your money. Okay. So what's going to happen is it's selective. It allows certain molecules to come in and certain things not to. You need to be able to get ions and nutrients into a cell, and you need to get waste out. Cell products as well, like a cell, for example, a gland cell makes a secretion. We need to secrete it, okay? A product. Breakdown of nutrients and production of energy is something the cell membrane does. ATP is produced. The mitochondria itself uses its membrane, its inner membrane, to make a lot of energy. It also does structural support and anchors the cell and tissues. So one of the things that we can do is we can take these proteins on the outside of the cell membrane and integrate it into the extracellular matrix and help attach. We can anchor cells and tissues. Remember in A&P we talked about cell junctions. We talked about things like gap junctions, occluding junctions, tight junctions, okay? Macula adherens, desmosomes, that kind of stuff, okay? The cell junctions that we talked about in A&P. Now, what's going to happen as well is we can transport across the cell membrane. Transport comes in two types, active and passive and active, okay? Now, the very important thing about it first is passive does not require energy. That's what makes it passive. It just goes with the flow. Next important thing about it is it goes from high to low with the flow. So it goes from high to low, okay? Passive requires no energy and will always go from area of high concentration to low concentration. Active transport, and that should say can, I said a cab, sorry for that minor typo, I am going to save it. I try not to, I try to be perfect, but I'm not perfect. Never been perfect. Okay, but if I don't change it now, I won't remember to fix it. So anyway, I apologize for that. People complain sometimes I do that. If I don't do it, I won't remember to fix it. So active transport is the only one that can go from low to high. And it uses ATP, mostly ATP, but we can use electrons, okay? Electron transport chain, all right? Now, it can go against the gradient and move from high to low, okay? Now, let's start with diffusion. Diffusion is, first thing we have to understand diffusion is to understand that molecules move. Molecules are always in motion. Now, molecule movement is based on things like temperature. The hotter it is, the more they move. But molecules are in motion. And with this motion, they tend to move. Now, what happens because they move? They spread out. They diffunder. Okay? They spread out. What happens is molecules go from area of high to low concentration. They spread out. It will diffuse down its gradient. And what will happen is it will go from high to low. They spread out, and it's diffusion. This spreading out is referred to as being a passive process. Now, what happens is it goes from high to low. What happens is, guys, as it spreads, it can spread out to where it's even across everything. We call this equilibrium. When particles on both sides of a membrane or the particles are spread out evenly throughout the space, there's no more high to low to go. We call this equilibrium, so diffusion stops. Okay? There's simple diffusion, that's diffusion through a phospholipid bilayer, and there's facilitated diffusion, that's diffusion through a protein. Now, simple diffusion is when things that are hydrophobic go through the phospholipid bilayer, like a big steroid. Now, steroids are pretty big molecules, but they are lipid soluble, so they're nonpolar. So they go through a cell membrane relatively easily, but if you had something that was large and polar, it won't fit into a cell, can't, without going through a protein. Okay? Facilitated diffusion is passive transport of anything non-lipid soluble, hydrophilic things, that must go through a protein. Anything with a charge must go through a protein. Anything that is uncharged can go through the phospholipid bilayer itself. Okay? So simple diffusion, basic diffusion here. Here we have some dye. Okay? Maybe this looks like some potassium permanganate. That's what this looks like. We drop it in and it dissolves and the molecules spread out. And as the molecules spread out, it eventually will reach equilibrium. Simple diffusion through the phospholipid bilayer. Facilitated diffusion through a protein. Okay? Now we're getting ready to talk about osmosis. Now, to understand osmosis, we have to understand something here. This is movement of water, diffusion of water. The diffusion of water is called osmosis. Usually osmosis happens to an aquaporin protein, but water goes from high water to low water. And you're probably asking yourself, how can I have high water? Let me help explain this to you. Okay? Let me help you understand what do we mean when I say high water or low water. Okay? Now, real quick, let's say I have NaCl. Okay? Sodium chloride. And I put this sodium chloride in water. What will happen is it will ionize. It will dissociate into Na plus and Cl minus. It dissociates or ionizes into these ions. Now, what does that have to do with water? Here, let's have a water molecule. Okay? Here I have a water molecule. There's the oxygen. There's a hydrogen. There's a hydrogen. Now remember, these hydrogens are partially positive. Okay? The oxygen is partially negative. Water and big white bears are both polar. Water being polar has a positive side and a negative side. Now what's going to happen here is, guys, opposites attract. The Cl minus is going to interact here with the hydrogen. The Na plus is going to interact here with the negative. And what's going to happen is other water molecules trying to write hydrogen in there, but I wrote it a little bit too small. They're going to surround that. Okay? And what's going to happen here? Other water molecules are going to surround this. Okay? Alright? So what's going to start to happen is water molecules are going to surround chloride. Water molecules are going to surround sodium. Sodium and chloride want to form an ionic bond. But now they can't because water is surrounding them. What has happened here is water is preventing these two ions from forming an ionic bond. So what's going to happen is water surrounds it, keeps it surrounded, and isolates them. So anytime that I ever increase solute, that will mean that I have decreased water. Okay? So what happens is, guys, do we understand why that is now? Not just that it happens, but why. We understand. We don't just memorize. Now what we're going to see here is when water, soluble things are added to water, some of the molecules are used to keep these things dissolved. This lowers the amount of water. We call this the water potential. Lowers the amount of free water. So when solute's high, water is low. There's less free water. When solute's low, water is high. There's more free water. Okay? Alright. Now what happens is you get this thing called osmolarity. This is how much stuff is dissolved per liter. There's 300 milliosmoles per liter of things dissolved in your blood. Tonicity is the ability of a solution to cause your cells to lose or gain water. That depends on the concentration. Always remember that osmosis, it's a real hoot. Water always moves towards a greater solute. Now, what happens is osmotic pressure is the force generated in pure water moves into an area of higher solute. When water does osmosis, that creates a force called osmotic pressure. All right, let's understand these terms here. Okay, what am I going to do is I'm going to talk about it like this: iso, hyper, hypo. Okay. All right, now let's get some beakers. Okay, let's say these are beakers. Okay. And in these beakers, I put some water. Okay. There's water in these beakers. Then to these beakers, I drop a cell. Little red blood cells dropped in there, okay? Now, these cells maintain homeostasis. So let's say the cell stays very stable at having this osmolarity. Okay? Very stable osmolarity. The cell doesn't change. But the world around it can. Now, let's just put 3 in that one. Okay? I'm going 3 to 3. Okay? Now, really, this would have to be scaled up ratio-wise. Okay? Because there's more water on the outside, we'd have to change the ratio, but it's alright. I'm going to do 3 molecules here, 3 molecules here. Okay? So you can see they have the same concentration. Okay, now we call that isotonic. Now osmosis, it's a real hoop. Water always moves as a greater solute. Now, where is the greater solute? Nowhere. It's equal. So what's going to happen is water neither moves in nor out. There's no net fluid movement. Now let's say on this one we have 1, 2, 3, 4, 5, 6. 6 solutes. We've really increased the solute concentration. Okay? This one here has a high solute. We call that hyper, which means high. Hypertonic. Okay? Now, because these solutes are hydrophilic, water will move towards the greater solute. The cell will shrivel up, because water leaves it. Water will leave the cell. Okay? Now, let's say this one here only has one solute in it. Osmosis, it's a root. Water always moves towards the greater solute. Water moves in and the cell swells. Okay? We call this environment here hypotonic. Okay? Alright. I'll let you pause there if you want to discuss it. Okay, now, in an isotonic solution, the cell's equal concentration inside the cell and outside the cell. Water moves in and out equally. There's no net movement. Hypertonic environments, the cell's higher concentration is in a solution. The solution around the cell is higher. This makes the cell low inside. Water moves out. The cell can shrivel up. We call this hemolysis. In plant cells, we say they plasmolyze. Plasmolysis is actually going to cause your cell membrane to strip off the cell wall and detach from the cell wall, and that can be fatal. Hypotonic environment, the solution has a cell lower concentration outside than in. Water moves in, the cell will swell up, it could burst, we call that hemolysis. In plant cells, it's just turgid. Okay? Now, because bacteria have cell walls, they aren't going to burst so easily. Bursting is a little harder for it to do, but the cell wall is weak enough, it can swell and it could burst, but the cell wall prevents that. So here you can see this baggie has sucrose in it and the world around it doesn't. So what happens is water will move in to make this baggie swell up till it reaches equilibrium. Okay? Cells, bacterial cells, might lyse if they're in a hypotonic environment. The cell wall gives it a little bit of protection. Now we're in the bacterial cell again. I said most of this would be on eukaryotes, but there's a little bit of prokaryote. But I wanted to stop before I got into all this stuff. I thought about, I was trying to find the perfect place to stop the last time. Now, the bacterial cell structures, we have our cytoplasm. That's the stuff inside. It's like 80% water. There's proteins, lipids, carbohydrates, and ions and stuff in it. It's a soup. Our nucleoid is the region where the chromosome is. There's a bacterial chromosome, usually very single. It's long but continuous in a circle. Frequently circularly arranged. It is double-stranded. People seem to think that bacteria don't have double-stranded DNA. All DNA is double-stranded. It holds the genetic information, but there's no histones, there's no nuclear envelope because these are prokaryotes. There could be plasmids, which are extra little bits of DNA in them. They are not actually, the plasmids are not associated to the main chromosome, but they are going to be proteins. So what I used to do in the lab is I would take a plasmid that we would generate. We would put a jellyfish gene of green fluorescent protein on it. Then we would put an antibiotic resistance gene on a plasmid. We would take that and make the bacteria take the plasmid up. And what we would do is our bacteria would glow in the dark. Okay? Alright, now ribosomes. When we deal with ribosomes in prokaryotes and eukaryotes, which these are prokaryotic ribosomes. How do I know? I'm going to tell you why. Ribosomes are little protein factories. What they do is they take an amino acid, amino acid, and make long chains of amino acids. They're made of two things, one called a large subunit, one called a small subunit. There's protein and ribosomal RNAs in it. Now the small subunit, in this case, is a 30S, and the big subunit's a 50S. Now wait a minute. 50, 60, 70, 80. Wait a minute. 50, 60, 70, 70S ribosome. What? It's a Svedberg unit. This S stands for Svedberg unit. And the Svedberg unit is a sedimentation unit. It is not a number we're adding together. The small is 30, the big is 50. Makes sense. Put them together and it's 70. Now, what in the world is going on here? It's a sedimentation unit. The small subunit, if I centrifuge it down, sediments at a unit of 30 Svedberg units. The large subunit by itself sediments at a unit of 50 Svedberg units. Put them together, and they sediment at 70 Svedberg units. Okay. Together, they sediment differently than they do separately. This will be on the test. I'm going to ask you who the small and large subunit are. 50, 30. Okay? It adds up to 80. That's 70. They're smaller and less dense. Eukaryotic ribosomes are ADS. Something else we might have is inclusions. There's lots of things we can do with inclusions. They can be storage of nutrients. They can be used for potentially. So maybe they store. I've always had this idea that some of them might be almost sensory. They do buoyancy. I've always wondered if any of them are for anything else, any sensory abilities whatsoever. If they do any sensation chemically. So that's just an idea that I've had deep in the back of my mind. But to my knowledge, there's no evidence to support that. So we have what's called a metachromatic granule of elution. They're just phosphate reserves for ATP. There's polysaccharide granules. That's a bunch of glycogen and starch. There's lipid inclusions for lipid storage, or sulfur granules for storing sulfur to make amino acids. There are carboxysomes, carbon dioxide fixation, because that's important for some of the chemistry that plants do, and also, some of the chemistry of photosynthetics, and some of the chemistry these cells produce CO2 as a byproduct, just like you do. Gas vacuoles for buoyancy, and even what's called magnetosomes. Magnetosomes have iron oxides in them. I've always wondered if they had any ability to do any sensation. I've always wondered that. It's a weird thing to think about sometimes, but I do. Okay, now endospores. Endospores are these weird things that are very resistant. They're resting structure found inside some bacteria. Some bacteria produce endospores. When the bacteria is like, wait a minute, I'm getting hungry, it make an endospore. It's released into the environment, very resistant to drying, heat, and chemicals. Two that really produce endospores, very well known for this, is the genus Bacillus and genus Clostridium. They can sporulate where the endospore forms, especially when carbon and nitrogen gets low, and they can germinate and return to a vegetative state. They can remain dormant for thousands of years, and then when the endospore coat gets damaged, they can germinate. This is weird. Like, we know that thousands of years, we've been able to find stuff that appears it was thousands of years old, and they reanimate, come back. Now, so I told you we're going to focus mostly on eukaryotes in this section. So, not going to lie, we did a little bit of prokaryotes. Now let's focus on eukaryotes. Eukaryotic cells like a complex city. I like to think of it like a city. And each building in the city has a function. Different parts of the city. So like there's like a city hall, there's like a power board, there's a waste management, there's a major office, there's warehouses, all kinds of cool little things. There's roads and highways and byways. Now, I don't like that they do half plant, half animal. I don't like that. I do not like it whatsoever. A flagellated cell that has chloroplast bugs me to no end. Hate it. Okay. Now, cilia and flagella. These are structures that might be on microbes, especially, and on some eukaryotic cells that aren't microscopic. It's not like sperm cells. Okay. Cilia and flagella have an ultrastructure that's the same. They basically have this sheath of plasma membrane and microtubules in what's called a 9+2 arrangement. There are two central tubules right here, and then there's 9 in a circle around it. 9+2. Okay? The basal body that anchors a cilium or flagella has a 9+0, like centrioles have in a eukaryote. They also may have motor proteins like dynein and kinesin, which bend the flagellum and cilium that allow it to swimmy. There's cell walls and glycocalyx. The cell walls of eukaryotes are simpler than that. Prokaryotes, they are found. Cell walls in plants, animals, and fungi. The carbohydrates, chitin and cellulose, are used to make those up. A glycocalyx, carbohydrates in animal cell membranes, it's like proteins and lipids with sugars on it. Okay. The cell membrane in eukaryotes and prokaryotes are similar enough. There's differences in proteins. There's differences in a few other things. The eukaryotic membranes have carbs. That's really good for attachment of things. They can have sterols to resist lysis and maintain fluidity. There's no group translocation in eukaryotes. Eukaryotes do endocytosis, like phagocytosis, cell eating, pinocytosis, cell drinking. I always kind of remember pinocytosis. I think of a Pinot Noir or Pinot Grigio wine that you might drink. Okay. Cytosol was the fluid inside. Okay, it's the fluid. There's substances in the plasma membrane outside the nucleus. We call that the cytosol, the fluid part. There's the cytoskeleton, and that's all of the microtubules and filaments, microfilaments, intermediate filaments, microtubules that makes the skeleton of the cell. It helps move in the cell and transport cell. There's a thing that's really important, especially in plants, called cytoplasmic streaming, where they basically move the cytoplasm around like a current, called cytoplasmic streaming. And this is movement of the cytoplasm around the cell inside, called cytoplasmic streaming. Now, we talked about how ribosomes are different in prokaryotes and eukaryotes. In prokaryotes, there's 70 S with a 50 and a 30. That makes 80. Now, 60, 70, 80, 90, 100. Same problem. This is a sedimentation unit, not a size unit. The large subunit sediments at 60 Svedberg units. The small subunit sediments at 40 Svedberg units. Together, they sediment at 80 Svedberg units. Again, these are sedimentation units, not size units. There are two different kinds of ribosomes when it comes to eukaryotes, free and fixed. Free are floating around the cytoplasm, and they make any proteins for the inside of the cell. Fixed are stuck on the nuclear envelope and the endoplasmic reticulum, and they make proteins that are going to be produced, secreted, or for the lysosome. They're going to make products that we secrete. Okay. Centrosome has a 9+0 array, kind of like the basal body. Okay. There are 9 triplets and no triplet in the middle. Okay. There's what's called a pericentrolar area, and there's the pericentrioles. Okay. They're involved in making mitotic spindles. Okay. Now let's talk about some organelles that are really big, important. They're structures they have with very specific shapes, very special functions. They are separated from the cytoplasm. They're called membranous. If you're membranous, you're separated. Now, what I like to say, here's kind of like a preface. I like to talk about these. I like to talk about the different organelles. I said the reason why we have different organelles and we have them membranous is this. It's like why your bathroom is not, you don't poop in your kitchen. Because the stuff in your poop is stuff you don't want to get in your food. You don't cook in the bathroom. You don't sleep in the kitchen usually. What we do is we have different rooms for different functions. Same thing goes for the cell. The chemistry of the Golgi may be interfered with by the chemistry of the mitochondria. And we don't want those two mixing. So our nucleus, our endoplasmic reticulum, our Golgi, our lysosomes, our peroxisomes, our mitochondria, and chloroplasts are some organelles we're going to talk about. The nucleus is by far the thing you're going to see the easiest if you look at a microscope. If you look at a microscope slide of a eukaryote, the nucleus is going to stick out. It's the biggest one, the biggest organelle that's easy to see. Okay? It's the control center for all the things. It is like City Hall. This is where all of the plans and all the things are there. All the buildings in there. Here's where we actually keep a copy of the blueprints to make all the DNA. It determines your cell shape, your structure, protein synthesis. It's the molecule of heredity is found here. DNA is found inside your nucleus. There is chromosomes in the eukaryote. In the eukaryote, there's a nucleolus. That is the nucleus of the nucleus. That is the center of the nucleus where ribosomal RNA is made, rRNA. There's chromatin. That's DNA before division. There's a nuclear envelope to isolate the nucleus. There's nuclear pores to let things come and go through the nucleus. There are histones. Histones are found only in eukaryotes. They are proteins that the DNA molecule wraps around. Nucleosome, which is made up of these histones and DNA wrapped around it, is unique only to eukaryotes. Prokaryotes won't have a nucleosome. They won't have histones. Histones are found only in your eukaryotes. Okay? Now, the endoplasmic reticulum, okay, is going to be a bunch of little tubules called cisterna. The ER is continuous with that. It actually connects to the nuclear envelope. Okay? There are two kinds, rough and smooth. The smooth lacks ribosomes, and the rough has ribosomes studded all over it. The smooth ER lacks, we call it smooth. Okay? Their functions, the smooth ER, in the synthesis of lipids like steroid hormones, sex hormones, metabolizes carbohydrates, detoxifies your drugs and poisons, and stores calcium ions for the cell. The rough ER, which is covered in ribosomes, fixed ribosomes, and it's rough, it's going to receive any proteins, any polypeptides made by the ribosome, it gets it. It actually helps them fold up into their structures, the secondary and tertiary structures, and many of them are packed in the vessels and sent to the Golgi, and the Golgi sends it out to be secreted. Transport vesicles are like the pickup trucks that hauls the proteins and things like that, deliver their stuff, and it goes to the Golgi. The Golgi apparatus is going to be like the post office. It's what arrives and ships, so it's like a post office. There is two faces, a receiving face and a shipping face. A cis face and a trans face. Now the Golgi has about 5 to 6 folded membranous sacs called cisterna. Looks like dinner plates. The cis face or receiving face is on the same side of the ER, and the trans face or shipping side is on the opposite of the ER. Vesicles leave this trans face and goes out to the membrane to be sent out, okay, to the world around it. Mitochondria, we probably heard this as the powerhouse of the cell. Mitochondria make energy from food to do work. The energy that it makes is ATP. It does that through a process called cellular respiration. It uses sugars and fats to make that. Now, what's cool about your mitochondria is it's got an outer membrane and an inner membrane. We think this is basically a bacteria that engulfed by another cell and stayed there and became symbiotic with it. It's called the endosymbiosis theory. That we evolved it. It was a collaboration between another cell. So we have this thing making energy. It's a bacteria. It has a lot in common with bacteria. Circular DNA, cellular respiration is done the same way as bacteria. Enzymes work the same way. It's got an inner and outer membrane like gram-negative bacteria have. Things like that. The outer membrane is smooth. The inner membrane is highly folded. And the inner membrane is going to be studded, what we call protein-rich, studded with enzymes. Now, these folds that it has looks like a rooster's crest. The word crest is crista. The fluid inside of that is called the matrix. In the inner membrane is our matrix. So between the outer and inner membrane is an inner membrane space between the two. Okay? All right. Now let's talk about peroxisomes. They are special compartments that have hydrogen peroxide in it, H2O2. They're basically, they use oxygen atoms from peroxide to break apart lipids and detoxify things like your body. They have enzyme catalase, very important for us, catalase positive bacteria for the lab stuff a little bit. We'll talk about it in lab. It won't be as big for you guys because you're online, at least this summer. I'm hoping by fall to have that different, and it'll be a bigger thing for my fall students. Vacuoles. These are storage things. They're food vacuoles and contractile vacuoles. They are phagocytosis happens and things are brought in. And food vacuoles are like what we store starch in plants, versus contractile vacuoles is what some protists use to maintain water balance. Chloroplast is going to contain chlorophyll. These are the things that are used for photosynthesis. They're found in the leaves of plants. There's an outer membrane and inner membrane. Inside, there's a stack of things called a thylakoid. The thylakoids, there's a granum, which is a stack of thylakoids. And then around the granum is a stroma, which is fluid. Connecting the different stroma is the stroma lamella, and connecting each granum together. And the stroma is the fluid around the thylakoids. Very important when it comes to photosynthesis. There is a list of some of the things that we saw in bacteria. Some differences and major organelles and differences. Okay? All right, guys. I'll see you in the next one.